So we were talking about whether these all hydrogen containing substances are acidic or not, and this was the activity. There are two iron nails in the solution and they are connected to a battery and then a bulb, and the bulb glows in case of HCl, HNO3, H2SO4, etc., but it does not glow in case of sugar and etc. So the sugar, whose formula is C12H22O11, this also contains hydrogen, but this hydrogen is not in the form of H+. It does not dissociate as H+, and as a result this whole sugar does not dissociate at all. So there are no ions formed, and that is why the sugar does not conduct the electricity. Alright, so now we come to a very interesting thing, that suppose you have a test tube and you have H2SO4 in the test tube. So this is H2SO4, and you add to it some sodium chloride. So you add some common salt to it. So there will be a reaction, and what will be that reaction? This will give Na2SO4 and HCl, just changing the radicals. So this is balanced. So this HCl will be in gaseous form, and this HCl will actually evolve from the test tube. But this HCl in gaseous form is dry. What do you mean by it is dry? Is that HCl, when you have a solution of HCl, you have hydrogen chloride and you have water in that solution. But in this case there is no water, only hydrogen chloride is evolving. So this hydrogen chloride, which does not contain any water, is called dry HCl. Now this dry HCl, if you want to test, you bring a blue litmus, and we know an acid changes blue litmus to red. But you will see that the color of blue litmus remains unchanged. Now we all know acid turns blue litmus red. Then why does the color of the blue litmus remain unchanged? It should become red. The reason to that is that what does litmus do is that litmus will not test the acid. Litmus, it only checks the H+ ion present. And since acids liberate H plus ions, it actually checks the acid. It is a test for the acid. But in this case, the HCl is dry. So, and we have seen that the litmus does not change color. So naturally, in this case, the litmus, there should be no H plus ions generated, because the litmus only tests for H plus ion present. If there is H plus ion present, it will change the color. So in this case, there must have been no hydrogen ions generated. So this dry HCl is not actually generating H plus ions. Now how can we say that? An acid, we have learnt in the first place that acids are substances which generate H plus ions. So this HCl, which is the very popular acid, hydrochloric acid, how can it not generate H plus ions? The reason is that the acids generate these H plus ions only in presence of water. This is very important. Acids will generate H plus ions only in presence of water. Now this HCl is dry, so there is no water, so this HCl cannot produce any H plus ions, so that is why the litmus test fails. But you take, now this one, you have used a litmus paper which was dry. The litmus paper was also dry. But if you dip the litmus paper in water and make it moist, when you have moist litmus and you have dry HCl, so this HCl will take up the moisture from the moist litmus, and then this moisture will act as the water for this HCl. And now since the HCl has got its water, now the HCl will generate your H plus ions. And these H plus ions, then again they will turn the blue litmus red. So in case of dry litmus, if you are asked to write the observation of this activity, you will write, in case of dry litmus, there is no change of color, and in case of moist litmus, there is change from blue to red.